Hello, everybody. David Sapirstein, Director of Business Development. I'm here today to talk to you about our Outsourced Pharma Capabilities update with SK pharmteco. For those of you who haven't worked with SK pharmteco yet, this is just a quick explanation of what SK pharmteco is in the broader understanding of SK. So SK is a global conglomerate based out of South Korea, and SK pharmteco is one of the 140 plus subsidiaries within within SK. SK pharmteco is primarily focused on the CDMO space for viral vector manufacturing, small molecule manufacturing, and peptide manufacturing. The goal of this call today will really be to review the viral vector manufacturing. However, if you have small molecule or peptide needs, please feel free to reach out and we'd be happy to connect you to the right teams. Here we see the structure of our facility layout. For viral vector, we have a facility in King of Prussia, Pennsylvania, just about 45 minutes north of Philadelphia. And we also have a viral vector facility based out of France. These facilities focus on AAV, lenti, and adeno manufacturing. We also have oncolytic virus manufacturing, which we'll get into in a little bit. One of the things I wanted to kind of speak to here is that beyond these viral vector facilities, we have a global supply chain for our small molecule and peptide as well. So we have facilities across North America, across Europe, and across Asia that kind of help support clinical trials wherever your clinical trials may be. So if you have a clinical trial running out of China, although our vector facilities are in France and Pennsylvania, we can support your clinical trial in China, Australia, wherever it may be. Here, this slide really denotes what we can do from a viral vector perspective. So what I want to do is kind of start at the R&D stage and speak to how we can support your AAV, lenti, adeno, oncolytic virus programs. If you come to us with a transgene that is problematic in any way, we can help you with your plasmid design and work through any of the problems that you may have. Let's say you're having an RCL issue. We can help support your plasmid design sequence to get rid of that issue. At that stage, we're ready to go through process development where we can start upstream and downstream development and scale your process for the needs that you need. Then we can get into the cell and viral banking space. So we have master cell bank and working cell bank capabilities along with master viral banking capabilities as well. And we also have our own internalized HEC293 cell line that we can support your programs with if you don't want to create a master cell bank, for example. At that point, we would move into the GMP manufacturing space and be able to support you from phase 1-2 all the way through commercial manufacturing with those analytical services internalized as well. So we have phase appropriate viral vector characterization that should be able to support you from the R&D position in which you start with us all the way through GMP manufacturing for, again, those AAV, lenti, and adeno services. One thing that we wanted to briefly call out here is our in vivo vector manufacturing services. Now we've had 3 or 4 different in vivo LVV programs that have come through our doors, and we feel like we've built out a really strong platform to support those manufacturing programs, whether it be in the process development space or even regulatory support. That's just one more thing to note for AAV, lenti, and adeno is we can support your regulatory filings across IND through BLA filings as well. So this kind of generally encapsulates what we can do from a viral vector standpoint. And if you want to bring in your own process, we can probably support it through tech transfer, anything that can come through our doors, we're able to support from a viral vector standpoint. But we also have what we're very excited to announce, our SkyDeck viral vector manufacturing platform. This is a flexible, harmonized approach of our viral vector services that we have been going on for the past few years, right? So we've been working on viral vector services for AAV, lenti, adeno, other oncolytic viruses for some time now. And this is really a harmonization of those services, right? So again, you bring in your R&D plasmid and say, hey, we just want to get to clinic as quickly as possible. We can turn that around from transgene to clinic in around an 8-month time frame. And this platform really enables us to do that. At the same time, we recognize that not every single vector comes to us in the same way, and not every single product and indication has the same needs. So what our SkyVec viral vector platform really enables us to do is branch into these different options that we will get into in a little bit. So from a GMP perspective, what has enabled us to build this platform is over 100 plus viral vector GMP batches. We can go up to 1,000-liter scale and suspension. We have 500-liter adherent batches and 9 different vector suites across those King of Prussia and France facilities that we shared earlier. So to dive a little bit deeper into what it is that we do on the SkyVec viral vector platform, we're going to focus on AAV, Lenti, and Adeno. Here is our AAV portion of our SkyVec viral vector program platform. And what I really want to focus on here is it's your typical HEC-293 cell line with a 3 plasmid system. We've worked across various different transgenes, all of your typical AAV 1 through 9s and novel capsids as well. And again, what we're really trying to show here is the flexibility in our platform. It is not a rigid approach where you come in with your transgene, we only scale up in one way, and this is what you get at the end of the day. What we really want to show here is that from a titer perspective, we can operate in the GMP space at a 50-liter scale. We can operate in the GMP space at a 500-liter scale. So I want to quickly walk through a few scenarios with you that we have experience and why our AAV SkyVec platform really makes sense for the clients that come to us. So in scenario 1, we have a well-characterized, established transgene that's come over to us, and somebody says, we don't have any rep cap, we don't have any plasmid build out, we just have this transgene for this indication we want to go off of. We've seen programs like that turn around in around 8 months to clinic, and you are already in clinic, you have your tox material made, and it's ready to go. The other scenario is, what if you're going after an indication that's ocular or CNS, where you want higher purity and probably don't need as much or much titer because it's a rare disease program? We are happy to scale and go up to a 50-liter in the GMP space to support your program and optimize the downstream in a way that you're getting that 90% full capsid when there's maybe some trade-off with how much titer that you get back on that other end. The last scenario from an AAV perspective that we've seen come through is for an indication that's cardiovascular in nature, where it's IV infusion. In that scenario, maybe we need to scale up to a 500-liter scale with our platform and get you as much VG as possible, right? So the point here is SkyVec is not a one-size-fits-all. We can approach your AAV needs in any which way. And real quickly, before moving on to Lenti, if you look over at that chart on the right-hand side of the screen, that shows the scalability in action, right? Where we start at that small 250 milliliter scale and then go all the way up to the 500-liter scale. We see that scalability from a titering perspective consistent across our platform. So whatever your platform or whatever your program needs are, we're able to approach it with our AAV SkyVec platform. So after reviewing the SkyVec AAV, we have our SkyVec Lenti offering. And what the SkyVec Lenti offering is really a culmination of 40 plus GMP batches and 20 plus transgenes over the course of the last 5 or 6 years. And we were able to create an adaptive approach really in the last 3 or 4 years because we saw the market shift from a high ex vivo need to a higher in vivo need. And we thought to ourselves when creating this platform, why would we build a system that is only built for one lentiviral vector approach? There's multiple different vector designs that we kind of talk about on the left side of the screen, but we wanted to design an approach that would work for all Lenti offerings. And so here you see our ex vivo Lenti workflow, and that focuses on our upstream manufacturing side of things, clarification, TFF, as well as additional purification steps. What we want to be clear about here is if you bring in ex vivo work to us, we'll continue our ex vivo workflow. That's what makes sense. We're not going to set up a system that is not, financially sustainable because we've only set up our Lenti system for in vivo processes now. And what I'm really getting at here is those 2 steps in between are focusing on our in vivo needs for in vivo clients, right? So for in vivo, the step that we are including is the seco endonuclease step, as well as a additional polishing step. And the goal for that is to get drug product level purity, potency, and characterization that is sufficient for the needs of an in vivo program for direct administration, right? So the percent recovery, the percent clearance that we're seeing when adding these steps is at a higher level than ex vivo programs ever need. So again, the SkyVec platform is really a branching build for these different vector programs and the needs of your program itself. So again, when you think SkyVec Lenti, think we have an ex vivo workflow that should work for programs that may not need that high purity. But if you're an in vivo program, we've worked on 3 or 4 different in vivo clients at this point in time that we have built out this additional downstream purification process that works from a clinical perspective and with the agencies. So now we're going into our SkyVec Adeno approach. Again, what we want you to think about is this branching approach on the SkyVec platform, right? Where we recognize that not every single adeno vector is built the same way. We recognize that there's, the traditional adeno approach, for example, which is a standard infection and that established regulatory pathway. We also recognize that for those larger payloads, a lot of people are taking a gutless approach with the helper plasmids. Um, and we understand that building out a platform that is only supportive of one of those approaches is a problem. So that's where the upstream process, we're allowing that to differ. It's kind of the inverse of Lenti, where from an upstream perspective, we recognize that there will be different approaches in the adeno space. However, on the downstream side of things, the innovations that we made for downstream manufacturing is what is really supportive of our platform, right? So we've replace that traditional heavy chromatography workflows, as you can see here, with a more streamlined hybrid approach. Again, when you look at those charts on the right, you're really seeing a similar concentration titer coming off with these different adeno approaches from an upstream perspective. What's that's showing is that we're getting a high level of recovery with our downstream approach that I believe is probably market standard, or sorry, not market standard. Let me replace that. Market competitive, and in fact, better than what we've seen with a lot of our competitors. We're getting a 70 to 85% total downstream recovery for adenoviruses. And I think that is really what, pulls SkyVec Adeno apart from other adeno platforms when we are understanding that adeno platforms don't really exist, right? This is a reproducible adeno platform that you're seeing with those 3 different charts up above. So in summary, going through SkyVec, what we want you to hear is that it's not one size fits all. We have a branching approach for your viral vector needs. We have, again, those programs that come to us that we can turn around in an 8-month time frame and get you into the clinic right away. But we also have programs that have very product specific needs. Those ocular programs that I've talked about, or auditory programs. We've had a few of those come through our doors where, you need a higher level of purity and different DP fills. We're happy to work with you for your program needs and how it can be used within our platform process. And I think the culmination of our SkyVec platform really emphasizes itself when we say we want to support rare disease innovators, right? I used those examples earlier, but we recognize that in the rare disease space, a lot of these broader platforms that people use where you have to scale to 500 liters or have to scale to 200 liters isn't appropriate from a financial perspective to make your rare disease program sustainable in the long term. So what we want to do as a partner with you is make sure that you have that financial flexibility, you have that process flexibility that works for your program. So we have launched our rare disease initiative earlier this year, and what we would like to ask anybody who's working on rare disease programs is let us help you and help your patients get the very much so needed drug that they need to support your program, but also support their lives and make it financially beneficial to both the company, us, and the patients in a way that we can, proceed forward for the future. Lastly, if there's any follow-up questions and you have any questions about our SkyVec platform as a whole, here's our QR codes. Please scan for the Lenti, AAV, and Adeno. If there's any questions or concerns, feel free to reach out to myself as well. And thank you for sticking with me today.